like everybody else. Hi, everybody. I'm super excited today to talk to you about protein large language models. I'm going to see here if I can get my notes up in speaker. Oh, here it is. It's really tiny. There you go. I'm going to tell you something that's probably not in my interest. I was taking the metro this morning and going through the Juno app and choosing talks to go to, and I realized that I had chosen like 3 that were during this slot. So I decided that I needed to create a sort of like clickbait tabloid style title to keep people interested and not going to other talks. Another alternative title is I used to hate PLMs, but this one weird experiment changed my mind. My name is Joseph Szymborski. Welcome to my hometown. I'm so glad you're all here. Make sure to come see me for restaurant recommendations if you need any. I'm a student at McGill University and MILA, the Quebec AI Institute, as well as a student in the Amin Ahmad's lab. I'm also a protein large language model skeptic, or at least a reformed one. You've all seen the title. Today, I'll be talking about experiments that have changed my mind on using pre-trained protein large language models for protein interaction inference. But I've gotten ahead of myself. My main interest is inferring whether 2 proteins interact given their amino acid sequence. These protein-protein interactions are often known as PPIs. We're in NetBio. I know everybody knows that, but I need to clear my conscience by saying that. In the past, we've trained a deep learning-based model to do just that, and we've called it RAPID. You can use that QR code to see the article published in Bioinformatics. Following up from that last talk, we do an analysis on the degree of proteins in our testing set to address in part the concerns in the last talk, but not in half an elegant way as described in the last one. RAPID works by first chunking amino acid sequences into tokens of 1 to 4 amino acids using a vocabulary of 500 tokens. We do that using a process called byte pair encoding, or BPE. We then input those tokens into regularized recurrent neural network architecture known as AWD LSTM. These encoders output a latent representation, and those latent representations are concatenated, and then that's sent to a classifier, which is a fully connected neural network, and that outputs a probability of interaction. That's all good, and that is that it is centered on recurrent neural networks, and that is decidedly old hat. The work I'm presenting today is spurred by comments I get from the community, as well as at conferences and my own committee, which went something like, why on earth aren't you using transformers or large language models? People are using them for everything. And I'm aware some people here may not know exactly what language models are. And so I'll go a little bit into that. For at least 2 decades, we've been trying to statistically model natural language using neural networks. And what that usually looks like is predicting parts of sequences using the rest of the sequence. So today we'll be primarily concerned with models which solve the masked language modeling, or MLM task, whereby parts of a sequence are omitted and the neural network is tasked with inferring the parts that are missing. So given a sequence, Montreal's blank are blank, a correct, properly trained model that doesn't make mistakes would say Montreal's bagels are best. So with the advent of the transformer architecture, the number of parameters in these language models have ballooned exponentially. And while those models focus on natural languages like English or French, many bright people, many at this conference, have been creating protein large language models, or PL, operate on amino acid sequences. And just for a sense of scale, the star down below is the method I previously published. That's RAPID. And as you can see on this logarithmic scale, it's very, very, very tiny. And these PLMs are really great at creating meaningful vector representations of proteins. Here we see a TSNE projection of the PROT-T5 PLM by the Ross lab, which demonstrates that embeddings cluster together according to their structure or lineage. It follows then you can create a twin neural network, just like RAPID, where the inputs are vector representations of proteins from a pre-trained PLM to leverage these meaningful representations for PPI inference. Here, 2 vectors from a pre-trained PLM are fed into a fully connected twin neural network, which serves as an encoder. The output from the encoder is then summed and inputted into a fully connected classifier network, which then outputs the inferred interaction probability. So why then have any skepticism about how appropriate it is to use these pre-trained PLMs as PPI inference models? Well, it comes back to what it always comes back to for me. We need to talk about the challenge in making generalized PPI prediction models. It's been known for quite some time now that it's a fraught process, and it's been illustrated as back as 2012 by Park and Marcotte. When we construct data sets for training and evaluating machine learning models, one may take a naive approach and randomly sample these interactions and assign them to training or testing splits. Park and Marcotte found that at that time, and subsequent papers have found similar things, that evaluation methods on the scheme do great across the board, no matter what they do. However, if what you do is then call a certain amount of proteins in these interactions training proteins and others testing proteins, and then remove any interactions between proteins that weren't of the same type, such that no training proteins are found in the test set and no test proteins are found in the training set, we find that the performance of those same methods degrades significantly, often dropping to near random. And this is true for deep learning methods. This has been shown by other groups in far more recent papers. So when PLMs are pre-trained, they use as large a training set as possible. That's usually all of UNIREF 90, all of UNIREF 50. And that means that the PLMs will be trained on proteins which are present in our PPI testing set. So even if we exclude testing proteins from our PPI training set, our PPI inference model still uses a pre-trained PLM, which has been trained on testing proteins. That means the majority of the parameters have been trained on these testing proteins. So naturally, the question becomes, are these pre-trained PLMs a source of data leakage for these PPI inference models? So we set out to test for data leakage. We just needed to train two PLMs, a non-strict one, trained as they usually are on everything, and a strict one, this time without any proteins in our PPI testing set. Just one problem. Training PLMs is hard. That's why you use pre-trained ones. Here we have the number of parameters on the y-axis and the encoding speed per amino acid on the x-axis, and both of them are on a log scale. And Rapid, my old model, is both fast and low on memory. So on my hardware, on my little 3090, it works fine. But PLMs are really big and slow by comparison, making them really, really hard to train. So we trained a much faster network we called SqueezeProtBERT. It uses an architecture called SqueezeBERT, which uses convolutional networks to speed up attention, and we just trained that on the amino acid sequences. So now that we've trained SqueezeProtBERT on both strict and non-strict data sets, it's time to plug them into the twin PPI network that I described earlier, that looks a lot like Rapid, and see if data leakage causes a difference in performance. And lo and behold, we observe no real difference in both between the strict and non-strict cases. What you see here is the average AUROC and Matthews correlation coefficient for PLM-based PPI inference. The numbers here are the average of 4 identical such methods that are using different random seeds. A reminder here that AUROC and MCC have perfect scores of 1. So top is best, and then the bottom is random performance. So now that we've pre-trained these PLMs and we know that they don't leak data, let's see how they perform. Compared to non-PLM based methods, the PLM methods perform much better as measured by Matthews correlation coefficient and AUROC, including our comparably small and fast SqueezeProtBERT model. So you can see here the diamonds are these non-PLM based methods, the circles are the PLM based methods. Each circle is a random seed, and the bar is that average of those. And you can see there's quite a big gulf there. So often, one thing I feel that I don't hear too often mentioned about PLMs is context length. So the complexity of transformers grows quadratically, at least in memory space, quadratically with sequence length. So while we may have proteins that are 2,000 amino acids in length, some PLMs operate on the first 512 amino acids. And so note that PROT-T5 here has the smallest context length, but if you recall from the previous graph, it performed the best. Also, PROS is actually based not on transformers, but on recurrent neural networks. So it doesn't really have a theoretical, it doesn't grow quadratically with sequence length. So it's not a huge deal what the length of the context is. So perhaps PROT-T5 is really good at predicting interactions between small proteins, but bad at predicting interactions between large ones because of that context length. But when we look at that, it doesn't look like there's any trend there either. Each bubble here represents a bin into which we've sorted testing interactions. The bins indicate the length of the protein sequence of each pair, with the length of one protein in the pair on the x-axis and the other on the y-axis. The more red a bubble is, the better the model predicts the interactions in that bin. The size of the bubble indicates how many testing edges are in that bin. So if we thought there would be like some length-dependent thing going on, we'd expect the bottom left to be very red and the top right to be very blue, but we don't really see that. So it seems that PLMs are good at everything, but maybe they're not. There are some things that we noticed that they didn't do as well as we had hoped. So for example, we were interested in seeing how well these models would do in predicting interactions involving proteins that were very, very out of distribution. So one thing that we did was we took an interaction map between all the SARS-CoV-2 proteins and human proteins that was published in Nature, and asked how well these PLM-based PPI inference models were at making these predictions, or sorry, predicting these interactions. And it turns out that when you test these on human data, as we've shown in our, when we use the same models that we've been showing you all along, they perform basically random. Yeah, that success that we saw on the human data didn't transfer onto the SARS-CoV-2 interactions. Next, we want to see how sensitive these models are to small but meaningful changes in sequence. And to do this, we tested how effective models were at predicting binding affinity as measured by changes in Gibbs free energy, as denoted here as delta G. Here, we have two wild-type proteins. They have some protein binding affinity as measured here by delta G. And then we have, let's say, a mutation in protein A that results in a lower binding affinity or a greater delta G. The change in that binding affinity is measured by the difference of the change of Gibbs free energy, which is known as delta delta G. So just remember that delta delta G is the change in binding affinity here. A delta delta G greater than 0 means that the interaction requires more energy to happen, whereas the converse is also true. So we can also measure the change in inference. We could also measure the change in inferred interaction probability by our models. One thing we can do is we can infer the interaction between wild-type protein A and B, and we'll denote that as Y hat. And then when you have a mutation that decreases the binding affinity, you'd imagine that the probability of interaction would lower because the interaction is not as good. So we can call the difference of those inferred interaction probabilities as delta Y hat. So delta Y hat, or the change in inferred interaction probability, should maybe then be correlated with delta delta G if we have a well-calibrated model that behaves like we understand the biology. But in our testing, when we looked at all the methods, but here shown are the two best ones, we didn't really see any correlation at all, or if there was, it was very weak. So in conclusion, pre-trained PLMs don't seem to cause PLM-based PPI inference models to leak data. And this is a little bit egg on my face because people have spoken to me at ISMB in the past, may have heard me conjecture that this is the case, and I swear I'm going to prove it, and I didn't. I proved the opposite. PLM-based PPI inference models perform better than the existing methods that we tested. And it seems that there's, obviously challenges that remain. We weren't able to get these models to generalize to viruses using the approaches that we used here. And it seems that similarly using these approaches, the models didn't seem to be awfully sensitive to these small but important changes in sequence, which is not awfully surprising when you consider that they perform so well on a truncated sequence of 512 anyways. So thank you so much for your attention. If you want to learn more about my work or get in touch with me, get my email, you can visit the QR code on my website, and definitely check me out on Mastodon, which you should be on instead of Twitter. Thanks so much.
Thank you very much, Joseph. Are there any questions? Very nice presentation. Thank you. Regarding the differences in performances, so species can certainly be one element, but is there a difference in the test set that you use? Because you were using, I believe, string for rapid. So is there a
Yes.
comparison between string and an APMS data set here? Because they have very different in nature. So that might not only just be a species question, right?
Yeah, it's a great question. It is. I think of. I have two thoughts on that. One is we definitely do see that there's a modality. Modality is important to how these things are done. We've tested, for example, on BioGrid, which is much more similar to string than it is to the MS data sets. But we know that we have to like tune the confidence. We have to. It only works if the confidence is done similarly to string. The modality definitely matters, but in a way, that's what we're sort of testing. We're testing out of distribution protein sequences. The distribution here being string, not just species. You're absolutely right. Part of that effect definitely is probably due to it coming from MS. But we also know from other tests that species, even when you test within string, is a big out of distribution test for a lot of these things. But these PLMs are trained on all species.
Right.
So we do anticipate there to be, a bit of a match. So we know that MS doesn't get you all the way to 0 or 50% chance, and species doesn't get you to 50% chance, but SARS-CoV-2 does.
Right. I don't deny species playing a role here. And
Yeah.
that's certainly possible, especially with viral. But maybe looking at subsets of strings, playing with like certain aspects of string might help you to try to decipher what's going on.
Yeah, and just on that quickly, rapid in the paper, we do analysis per channel. And we find, for example, text mining, we predict much worse than, for example, co-expression or database. So yeah, there definitely is for sure, even within string. Yeah.
Thank you.
Thank you.
So following up on that, I have a quick question because it's what I was thinking, like the idea of these big models to have foundation that you would fine tune, and maybe finally your virus data are hidden in something. So do you think you could use this as a baseline and then try to fine tune specifically for detecting virus interactions?
100%. And we've shown similar stuff with rapid where we could fine tune and get great performance. But I'm always worried, especially with smaller data sets like virus data sets, that there's a ton of bias there. And it's really difficult for me to feel comfortable about the generalizability of our models when they fall flat on their face when the model comes from, albeit a very significant species. I just wonder, are we capturing biology here, or is it the networks effects and things like that? Yeah.
So thanks for using string. And a quick string-related question. You say you're trying the different evidence channels separately. Yes. Have you also tried the different evidence channels in the physical PPI mode? Because there are two modes now. There's the functional association network and the physical interaction network.
So we use V12 and we use the physical links data set.
Okay, so you use the different channels from that one.
Yes, exactly.
Okay, good. Then there's not co-expression in it.
Okay, good. Thanks so much. We love string.
I have one question for you. Nice talk. I wondered if you tried to interpret the resulting models at all to understand, like for instance, to discover domains and interactions between specific domains. There are good data sets that capture domain level interactions. You could use that to potentially understand and interpret those models.
It's definitely something we want to. I've definitely been interested in incorporating them more specifically. We have another model out called Intrepid, which uses orthology data, and we show that in that paper we show that there actually is, you can cluster the latent space, those latent representations, and we cluster by Go terms and uniproc keywords accordingly. But yeah, more specifically, using that like as actual like inductive bias, really interesting idea. Yeah.